Okay, let's just jump right in. What if I told you that for you to stay alive, your body has to purposefully kill billions of its own cells every single day? And no, it's not a bug or a malfunction. It's a feature. This is the incredible story of programmed cell death, our body's silent and absolutely essential process of self-destruction for the greater good.
It sounds a little strange, doesn't it? Talking about a good way to die. But on a cellular level, that's exactly what this is. We're about to see how this isn't a story of decay or failure, but actually a life-giving process that shapes us, protects us, and keeps us healthy.
You know, the philosopher Albert Camus probably wasn't thinking about cell biology when he wrote this, but wow, does it hit the nail on the head. For the whole organism, for you, to survive and thrive, individual cells have to be ready to sacrifice themselves when they get old, damaged, or are just no longer needed. It's really the ultimate act of teamwork.
So this process has a name, and it's called apoptosis. Now, this isn't some chaotic, messy death where a cell just explodes. No. Think of it more like a clean, orderly, and super tightly regulated self-destruct sequence. It's something evolution has been perfecting for hundreds of millions of years. It's a fundamental part of what it means to be a complex creature. So what's the point of all this cellular sacrifice? Well, it has some seriously important jobs. Apoptosis is the sculptor that carves out the spaces between your fingers and toes when you're developing in the womb. It's the body's quality control manager, constantly sweeping away damaged cells that could turn into cancer. And it's a frontline soldier, forcing cells that have been hijacked by viruses to self-destruct. It is, without a doubt, absolutely essential. But how in the world did we figure all this out? To understand how our own cells are programmed to die, scientists had to look somewhere pretty unexpected. A tiny, transparent worm that, believe it or not, held the master key to this whole biological puzzle. So, meet C. elegans. This humble little worm became the hero of our story for a couple of key reasons. One, it's transparent, so scientists could literally watch individual cells die right under a microscope in real time. And two, its genetics are way simpler than ours, which made it possible to hunt down the exact genes that were pulling the strings. And what's so cool is that you're seeing pretty much exactly what those early researchers saw. Those bright little white dots, the ones they described as looking like refractile, button-like corpses, those are individual cells in the middle of apoptosis. By just watching and counting these little lights wink out, scientists began to map the genetic blueprint for self-destruction. And what they found was just beautifully elegant. It wasn't some complex, messy web of signals. Instead, it was a clean, linear, four-gene pathway. You can think of it like a simple chain of command, passing the self-destruct order down the line from one protein to the next. Okay, so it all starts with a signal. A cell can't just decide to die on its own. It has to receive the order. And in the worm, that order comes in the form of a protein called EGL-1. Next up, that signal, EGL1, has one mission. Find a protein called CED9. Now, CED9 is the guardian of the cell. Its whole job is to keep that self-destruct sequence switched off. EGL's job is to basically tackle the guardian and take it out of the picture. And with the guardian, CED9 neutralized, well, the protein it was holding back, CED4, is now free. This is the point of no return. These CED4 proteins quickly clump together to form a structure called an apoptosome, which is basically the launch platform for the final execution. And finally, that platform activates the executioner, a protein called CED3. CED3 is a type of enzyme called a caspase, and you can think of it as a pair of molecular scissors. It just goes to work, systematically chopping up all the important parts of the cell until it's neatly dismantled. Simple, clean, a four-step cascade. So here's where it gets mind-blowing. That simple pathway in a tiny worm, it's not just some weird, quirky thing. As you can see here, each one of those four core worm genes has a direct counterpart, a homolog, in us. We use the same fundamental machinery, a signal, a guardian, an activator, and an executioner. But there's a plot twist. So while the cast of characters is basically the same, the way the story plays out in our cells is a lot more complex. Evolution added a whole new layer of control, a major plot twist that involves one of the most famous little organelles in all of biology. So, if we have the same basic toolkit, why isn't the process identical? What did evolution add to our system that the worm doesn't have? Well, it turns out a new gatekeeper was placed right in the middle of the pathway, and it completely changed how the whole system is controlled. And that new player is the mitochondrion. Yep, that's right, the powerhouse of the cell you learned about in high school. Turns out it's moonlighting. Its side gig is acting as the cell's execution chamber, holding the final key to life and death.
And this is the key difference. In the worm, the pathway is direct. Signal takes out the guardian, which releases the activator. But in us, the command has to go through the mitochondrion. It became the central hub, the command center, where the final decision to live or die is made. This slide here really breaks it down perfectly. In the worm, it's direct control. The guardian protein physically holds on to the activator protein. The signal just pries them apart. But in us, it's more indirect. Our guardian proteins act like sentinels on the surface of the mitochondrion. They don't hold the activator directly. Instead, they control whether a second signal gets released from inside the mitochondria.
And that crucial secondary signal is a protein called cytochrome C. Now, its day job is to help with energy production inside the mitochondrion. But if it ever gets out, if it's released into the main part of the cell, it is a death sentence. It immediately finds our version of the activator, APEF1, forms that apoptosome platform, and unleashes the executioners. Game over. Now, understanding this whole intricate pathway isn't just a fun academic exercise. This is where the fundamental science has huge implications for our own health. Because when this finely tuned death switch goes wrong, the consequences can be absolutely devastating. It is all about balance, a really delicate balance. If cells that are supposed to die refuse the order, that's too little apoptosis, they can survive and grow uncontrollably, which can lead to cancer. But on the flip side, if perfectly healthy and necessary cells start dying when they shouldn't, too much apoptosis, that can lead to neurodegenerative disorders like Alzheimer's and Parkinson's. And this is why this all matters so much. By understanding the exact molecular switches, the guardians, the activators, the signals, we can start to design drugs that can flip those switches. We can develop therapies that encourage cancer cells to die or protect healthy brain cells from dying. This basic research is the absolute foundation for a whole new generation of medicines. It's really incredible when you stop and think about it. The secrets to our own life and death, our health and disease, were first uncovered just by watching cells disappear inside a tiny see-through worm. It just leaves you wondering what other profound biological truths are hiding in plain sight, just waiting to be discovered in the simplest of creatures.